thanks everybody around the world who's tuning in. I'm here today with Dr. Gina Wimp from Georgetown. Hey, Gina.
Hello, Mark.
It is great to have you on MarshChat. Cool. So Gina is an awesome marsh ecologist, and we're going to talk a little bit about her research and marshes in general, and it's going to be pretty cool. So we'll just get started with an easy one, Gina. So you study a part of the marsh that not a lot of people look at, which are the terrestrial arthropods. In your humble opinion, what do you think are the most important terrestrial arthropods out there in the marsh?
Well, if we want to go strictly by number, I would have to say the plant hoppers. They're not as fun in terms of what they do. I mean, it's pretty simple. They just hang out and they suck on grass phloem. So that by itself is not that exciting. But what ends up happening is when we have nutrients coming into salt marshes, they can reach just absolutely insane densities. So they can number 25,000 per square meter and in Spartina alterniflora, and that's the Proclesia dolus and Proclesia marginata, the two main plant hoppers in those habitats. So just by sheer number alone, and they can actually, even though you don't see signs of damage like you do a chewing herbivore, they can nonetheless have a big impact on plant productivity in the system. Yeah. My favorite, I have to say, though, is the Pardosa littoralis, our hunting spider in that system. So those guys can move about 7 meters a day, and it's those generalist predators that really link this, what we think of as a terrestrial food web, to the marine food web for the arthropods in that system, because they will switch back and forth from feeding on prey that eat detritus and algae to later in the growing season. Once the plants get going and get established, they'll start to have an impact on those plant hoppers. And one of the reasons they can have an impact is that by feeding on that detrital algal food web early in the season, it can build up their densities, and then they can have a big impact later on. So really, when we're thinking about the terrestrial food web, there's a huge marine component to it.
Yeah, so that's interesting that you bring up the importance of nutrients and kind of generating this, because I mean we have examples, I guess. Well, we've got the examples of in the southeast of snails grazing on Spartina more when there's more nutrients. But I guess besides that, I mean, is it that and the Proclesia are the ones that respond so highly to increase nutrients? I mean, I don't think there's anything been done with like some of the other marsh plant eaters and nutrients. I don't know, can you think of anything else that fits into that?
Well, I was thinking actually that in some instances, David Johnson was seeing that it was actually going all the way up to the mummichogs.
Yeah.
But one of the things that we've noticed with some of these nutrient additions is that the group that responds most strongly is actually the predators. So what we see is that, I think that a lot of that has to do with the fact that they just have more prey and a larger diversity of prey. So you get higher species richness and higher abundance for these top predators. So a lot of the generalists, but also a lot of specialized mesopredators as well. And one of the things that we're seeing, and still this is very new, so we're just starting this work. One of the really unexpected things we had happen out in the marsh was when we started fertilizing, we just started seeing this dieback in these plots. And one of the things we started doing was, looking at that vegetation and sort of dissecting it. And what we're finding is that there's a stem-boring moth that's responding to that nutrient addition. And when those guys get done feeding, it's almost like a beaver went through and felled all of our culms. So that's one of the things that we're looking at as well, is the nutrient additions on that feeder that isn't going to be exposed to a lot of the other predators. It's going to be concealed in that vegetation. So nitrogen could be linked to dieback in that way as well.
That's really cool. So have you gone to marshes that maybe have different I don't want to say ambient nutrient levels, but there's like marshes that have different nutrient levels. Is there a difference that you can see in the grays? There's just kind of naturally, not manipulated differences in nutrients?
Yeah. So what we did in conjunction with this press pulse experiment, where we're looking at nutrient pulses, so just a single year of nutrient addition versus presses that have multiple years of nutrient addition, is we wanted to, and this is work with my collaborators, Shannon Murphy and Danny Lewis. And one of the things we wanted to see is, okay, well, we're doing this on a plot level, and really what does it look like when you go out into the world and look at marshes that have different levels of nutrient additions. And what we often did was kind of use the proxy that Burtness and Silliman did, where we looked at the extent of urban development near the marshes.
Yeah, sure.
And that correlated really well with amount of nitrogen in the plants in those systems.
Yeah.
And so what we did is we went from Virginia to Maine and looked at marshes that were fairly near each other, but as close as we could get, but had really different levels of nutrient inputs. And a lot of what we're seeing on the plot level is really mirrored on that landscape level as well. And one of the things that really sort of stuck out to me when we were going through these marshes, especially like when we went into Jamaica Bay, which is right next to New York City, is where you had really high levels, high amount of human activity and high nutrient levels, which we saw in the plants themselves. When you're walking through that marsh, even though you may have really tall vegetation, you've got a lot of these giant holes in the marsh. And so a lot of like what Linda Deegan has been talking about with the erosion of marshes due to nutrient inputs, like you could really see that when you got into those systems, because you would have like just bunches of culms, and then you'd have giant potholes where there was nothing holding the marsh together. So
that's really cool. Yeah, and so for those of you who don't maybe know exactly what Gina's talking about with the amount of shoreline development and stuff. Basically, along these different marshes, like if there's a woody vegetated buffer, those like shrubs and trees like suck up a lot more nutrients that are running out into the marsh. Interestingly, I would imagine that the amount of, say, shoreline or marshline forest probably affects the arthropod communities too, right? I mean, a lot of, I mean, I guess at what frequency are the arthropods in the marsh coming from the forest or moving back and forth?
There's really not a huge exchange there. There may be among predators, because a lot of these generalist predators can probably move across and do basically use these marshes as well as the forest. But what we see in the marsh is the two Spartinas are, in my view, if I was an herbivore, they would not be what I would pick. And they've got a lot of silica. They've got very little nitrogen. I mean, it's just, oh goodness, I just can't even imagine trying to eat those grasses. And so what we have is, for the most part, until you get down into the south where you start to see more of the grasshoppers, like where we are in sort of mid-Atlantic and moving north, most of the community is really specialized sucking insects. So they've got beaks that they're using to feed on the grasses. And what we find is when you have alterniflora, the lowland smooth cordgrass species, and then Spartina patens, the upland marsh hay, you have pretty much specialized herbivore communities on each of those grasses. And you don't see much of an exchange between them. There's really kind of just one delphacid plant hopper that we see in both areas. So if basically evolutionarily, if they've learned to feed on one, they don't know how to feed on the other. And so so that is one of the reasons why when we start to see Spartina patens getting more and more fragmented, it's not as though the herbivores that are found on that grass can just shift over to alterniflora. They're pretty much designed for for patens. They've got these really horrible wings so that they can move in that high thatch habitat, and they know how to feed on that grass. And knowing how to feed on that grass does not make it easy to feed on alterniflora. So
That's really interesting. So, I mean, kind of along those lines, can you maybe speak kind of quickly about like, or I mean, at any pace you want, about some of the different adaptations that that different insects that feed on different marsh plants have? Like, what's the difference between like what is it? Is it, you know, is it the length of the sucking proboscis that is different? You know, what sort of things are different between something that feeds on the smooth cordgrass Spartina alterniflora versus something that feeds on the thatchy Spartina patens, maybe versus something that feeds on, you know, juncus or something like that?
Mm-hmm. So one of the big things that, so this was Bob Dino and Andrea Huberty that they figured this out. One of the big adaptations we see is, and this is all Proclesio plant hoppers that are in alterniflora, is they invest differently depending on their feeding strategy and different things. So for instance, Proclesia marginata, plant hoppers do this crazy thing where they can either have one or two sets of wings. And that happens actually kind of a lot within the Delphasidae. So what Proclesia marginata will do is it invests in two full sets of wings and then moves from plant to plant looking for a high nitrogen host plant. Mm-hmm. And when you're actually looking at essentially the points of muscle attachment on the face of the plant hopper, they're very thin and it doesn't have tons of points of muscle attachment there. But when you move over to Proclesia dolus, most of the time it invests in one set of wings and will just sort of hunker down on whatever lousy host plant it has. And what it invests more in is essentially anchoring the musculature for the beak so that it can sit there and just throughput a lot of phloem until it gets the nitrogen that it needs. And so we definitely see changes like that in the plant hoppers in order to cope with those low nitrogen host plants. And you know, within the plant hoppers, they invest in different strategies all the time. And so the plant hoppers that we see within the patens, all of them have these shortened wings. So they just have one set of wings and it looks like somebody took scissors and just sort of cut them off. And so those guys, they live in a high thatch environment, and having two long sets of wings is there's just no need. And so those guys, not only them, but the specialized predators, these myrid bugs that feed on those plant hoppers. So they will go along and locate their eggs, which are embedded in the Spartina patens, and use their sucking mouth parts to suck the egg contents out. They even have shortened wings. Whereas when you move over into alterniflora, the specialized myrid bug that feeds on those plant hoppers has long wings. So you have these sort of mirrored communities, each with adaptations for living in those environments. But what's interesting and what we haven't worked with as much yet is that we're not sure how much the two different plant species are preferred by these different groups to just the feeding the adaptations, or also oviposition adaptations. We haven't looked into that as much. And when I was just at UVA last week, we got a really great suggestion from a person there about you should really look into it for ovipositing too, because those guys, they're actually ovipositing into the grass. So some of it could be that they just can't oviposit in the alternate grass. Yeah.
So do they feed and oviposit on the same grasses generally?
Yes.
Okay, that's interesting.
Yeah. So we want to maybe get into that a little bit and see really what's limiting them, because for each of those plant hoppers that just, you know, they're doing so well in their respective grasses, as you move toward the edge, toward the border of that alterniflora and patens, you get declines in the dominant species of both of the plant hoppers. And a lot of that, I think, has to do with their specialization. So even though there's no difference in the amount of nitrogen for plants in the edge, there's no difference in live biomass, there's no difference in thatch from the stuff that's growing in the centers of each of those habitats. those plant hoppers, all of the specialist plant hoppers decline along that habitat edge. So a lot of that could be, you can't make mistakes. And if you're in that area, you're likely to make mistakes.
That's really cool. So I mean, what you're describing here is really interesting because in general, when you look out at a marsh, you see, one or two, big monocultures of grass, right? Either the big Spartina alterniflora patches, big Spartina patens patches, potentially, some high marsh species as well. So you look out at a marsh and you think that, that's just like a bunch of grass out there. And yeah, there's some different species, but they're probably all being used by the same thing. But basically what yeah, it's just shaking your head. No, right? I mean, in fact, it's like, it's crazy specific, right? Like, I mean, so I know that, you've done, research along borders of these zones. Like, I mean, when we're talking about limited crossover, is it like, the only things that are crossing over are the spiders?
Well, yeah, mainly generalist predators, but not even all of the generalist predators will move, because here's the thing. I think people tend to think of generalists as always being able to negotiate these edges, these habitat edges, and live in fragmented habitats, no problem. And the thing is that people have to remember that not only do we have to consider their food. Now, they may be able to eat lots of different stuff, but habitat structure plays a large role in where these predators are distributed. So for instance, when we're in Spartina alterniflora, it's a very coarse, calmed grass that sort of stands up straight. And as you've seen with patens, it's floppy and sort of lies over on itself. So even though we have generalist web building spiders that can feed on organisms in both of those habitats and move their webs around in association with the abundance of prey, they cannot live any place other than the alterniflora, because the patens presents really lousy sites for web attachment. They've got to be able to anchor it. So they do that in the alterniflora. So even though we have these really specialized herbivore communities and everybody kind of assumes that generalists can just do fine anywhere, we do see that the differences in structure between those grasses really have an impact even on our generalist predators.
Yeah, that's so neat. I mean, because it's one of those things where, it's hard to, well, actually, I guess those of us who grew up with the Honey, I Shrunk the Kids movies, probably maybe those are good perspectives. When you get shrunk down to the size of an ant, then you can, the world is so different. I mean, so with these insects and these arthropods, they're so small. So yeah, I mean, being in that thatchy Spartina patens is so different than being in the alterniflora, right?
In fact, when we have 51 species of generalist predators that we looked at in a study across this habitat edge. And really 2 of them actually have a positive response to the habitat edge, which is what most people assume for generalists, especially generalist predators, because I think we've all grown up with the ideas of like brown-headed cowbirds and things like that, where you need habitat in one environment and you need food in the other. And so you see the generalists responding positively to the habitat edge, but it's a minority, just 2 out of 55, I think, species that responded positively to the edge. Every other group had a negative response. They were more abundant in one habitat and declined along the edge. So even our generalists basically can be limited by these habitat edges. And since they're one of the major factors influencing responses to fragmentation, then we need to think about them too. I think a lot of times we just assume they'll be fine, but apparently not.
Yeah, that's really cool. And I mean, I think one of the things that I know that I've noticed with different spiders' usage of different zones in the marsh is something that for me, you can see it a lot, like in the early morning when the grass is really dewy. You can see, and you probably didn't know which species do these different things. In the Spartina paten zones, you'll see the webs like that are basically like laying over the top of the grass, essentially, and so you can see the dew on them. And then when you walk into the Spartina alterniflora zone, you see the webs between the tall, the taller pieces of grass and stuff like that. So yeah, I mean, it's easy to see with a little bit of dew, you know, how just some different strategies in which I imagine are not super different spiders, like from a phylogenetic standpoint, right? Yeah.
Right, doing completely different things.
Yeah.
And one of the things that we see over and over again too is just because you're a member of the same functional group doesn't mean that you do things in exactly the same way.
Yeah.
Yeah.
That's very cool. Cool. All right. Well, I'm going to shift gears a little bit here, Gina. So for you, obviously, you love arthropods, but why is it important to you to study salt marshes?
Oh, salt marshes, in terms of their importance to humans and just as ecological systems in general to study, these guys, this system is so important ecologically and economically. And I did not grow up with these systems at all. My introduction to them was through Bob Deno. I grew up in the mountains, in the Rocky Mountains, and I'm about as far away from a salt marsh as I could get. But when I came out here in 2004 to the sort of mid-Atlantic, I started working with Bob. And it was amazing to me getting out into the salt marshes and really seeing the diversity that's out there, because everybody thinks it's a very simple system because you just have a couple species of grasses. But I think what people don't realize is all the things that are going on that are really hard to see. So the only way we can sample our communities is with this DVAC suction sampler, this crazy like 35-pound lawnmower we wear strapped to our backs, because everything is just embedded down there in the grasses. But salt marshes, they filter nutrients, they filter heavy metals, they act as nursery grounds for fish and shellfish. They're important in buffering the impacts of things like hurricanes on human habitation, and they're really important from a carbon storage perspective. And I think that they used to not get counted in that inventory because they're also releasing methane.
Yeah.
But what people have started to find is that the amount of methane that they emit is much less than the amount of carbon that they actually store in their soils. So since those soils are buried in those anoxic conditions by the tidal flooding, the plant material that winds up in those soils lasts for thousands of years. In fact, you can go through and use macrofossils and look at, which of the different species was in which particular habitat at what particular time. So Burtness has done some work on that. And you can really store a large amount of carbon over a long period of time. And I think they and the mangroves and other wetland systems, I think, that ends up being a much more important a system for storage of carbon than a lot of other systems. And one of the things we've seen with the nutrient addition experiments that we've done is that when nutrients come into that salt marsh, it actually increases the rate of above ground decomposition. So one of the things that we were really expecting is in plots where they had received fertilizer year after year, that we would just get this huge accumulation of dead material or thatch. And that wasn't happening. It was about the same as our control plots, or sometimes even less. And what was happening is that high quality nitrogen foliage was decomposing faster, but it looks like we're actually altering the microbial community, which is also speeding up the rates of decomposition, and providing shade, which speeds up the rate of decomposition, because then you have more moisture. It actually brings in more of these snails, the coffee bean snails, and those guys will actually shred the material, and their poop will actually stimulate the fungi. We get higher levels of fungi on that decomposing material. So all those things are sort of acting in concert to increase the rate of decomposition in those fertilized plots. So that has bearing on the importance of these systems for carbon storage.
That's really cool. Yeah, that actually preempted. Oh no, an echo on your side? It's just an echo. Anyway, well, that kind of preempted my next question for you, which was going to be we know about there. You've talked about the effects of these different arthropods on like primary productivity. The specific grazers. I was going to ask, are there any of them? Are there any of the arthropods that are affecting decomposition rates? So you just talked about how they kind of indirectly do. Are there is there data that talks about like the direct effect of these guys on turning over that dead material? Like, is there any which I guess the other question could be, what's the most important terrestrial arthropod decomposer, do you think?
Well, I would say coffee bean snail.
Yeah.
Yeah, yeah, easily the coffee bean snail. So I think that for our arthropods, a lot of them are probably causing the material to senesce a little earlier than it normally would be, which could be affecting the nutrient cycle. But by and large, those coffee bean snails are the big ones. Yeah.
That's really cool that the arthropods are having and the nutrients are having an indirect effect on that too. That's really neat stuff. Cool. Okay, so what do you think the future of kind of the terrestrial arthropod food web is with marshes? Like, what kind of threats does it face, and then maybe can we manage for it? I mean, that's a hard thing to manage for, I guess. But yeah, what do you think the future is with these with the terrestrial arthropod food web?
Well, I think one of the big things that we've been studying, of course, is the nutrient inputs, and like I said, that has a really strong impact on the upper trophic levels. That's not sustainable, because as you get more and more predators, they're gonna have an impact on the other members of the food web. And so really what we need to keep doing in the future is understanding more about the top-down impacts of those predators, not just on the live plant food web, but also on this algal detrital food web as well. I think one of the big things that's going to happen is the habitat fragmentation, and basically patents is just getting squeezed into smaller and smaller areas. And so what often happens, especially in our marsh, is when silt's deposited, it's sort of deposited into almost sort of the middle of the marsh. So it's not like patents can easily just move upland. So it's essentially being side. And as I was mentioning, pretty much the majority of the arthropods found in patents can't use the other habitat. So you'll start to lose those species. Actually, one of the things that we've been finding as well, and this is in conjunction with one of the other professors here at Georgetown, Matt Hamilton, and then our shared graduate student, Amy Botto-Clutty, is that fragmentation is actually impacting the evolutionary potential of some of the dominant herbivore species. So those declines we saw in abundance of our dominant plant hopper species and patents along the habitat edge, in association with that, we've developed microsatellite markers, and what we are finding is we also get a decline in effective population size. So it's not as though the plant hoppers from the center of the habitat and from the edge are panmictic. They're not basically coming together all that often for reproduction. And so we're seeing more higher levels of genetic drift along the habitat edge relative to the center. So what that tells us is long before that habitat ever gets so small that it just can't support the species, fragmentation is already having an impact because of those declines in plant hopper population size along the habitat edge.
Yeah. Wow, that's really cool. I mean, this is it's hard to convey exactly the scale that you're talking about here, but I mean, this is like, potentially, meters apart. There's huge differences, which is so cool, because I mean, this is a system that, at first glance barely varies at all.
Yes, exactly. Yeah. And the thing is, it actually presents sort of a model system for studying fragmentation, because oftentimes when we're looking at the effects of fragmentation on gene flow, if we're in the tropics and we're looking at trees that live for 200 years, we're not looking at what's happening to them currently. So these plant hoppers are great because they're short-lived species. Depending on which of the two dominants we're talking about, one has 2 generations a year, the other has 3 generations a year. So we can track what's happening currently, which is also the reason we're using microsatellite markers. We're basically looking at the current effects of fragmentation on those species. So it's actually kind of a model system for looking at that too, because those things are not coming from completely different gene pools from different regions or things like that. We can actually use them, use the species and look at fragmentation on a very tractable time and spatial scale to see the impacts on individual species.
Yeah, that's really cool. So something that occurs a lot in these sort of northeastern, and I guess basically from you on up, is the invasion of Phragmites. So this invasive, the Australian reed, I guess is what its common name is, right? What have you guys looked at what's going on with the Phragmites in the arthropod community?
So Claudio Graton did some work with this in our system, and actually at Alloway's Marsh. So it was Claudio and Bob Deno. And so he looked at basically the fact that the generalist predators were able to feed off of both food webs. At the Tuckerton Marsh, where we're working primarily, we don't have a ton of Phragmites. It's only along the road that goes out to the marine lab, and it really doesn't make its way that far into the marsh, which, thank goodness. But but yeah, at Alloway's, Claudio did some work on this to look at basically which species were shared between them. And again, there's not much that transfer in terms of the herbivores oftentimes, because what's specialized for alterniflora for one of the Spartinus isn't going to be able to move very easily over to Phragmites. Yeah. Cool.
Very interesting. Okay, Gina. Well, I'm gonna now start a new thing in MarshChat, which are Marsh rapid fire questions. So I've got 1, 2, 3. I got like
bonus round.
Bonus round. Yeah. So far you're doing good. You've earned a lot of points. You got a lot of Marsh points. But yeah. So just some fun Marsh related rapid fire questions. Okay. Cool. Okay. First, what is your favorite marsh?
Tuckerton. I've been to lots of others, and now I kind of understand. Whenever we went to a different salt marsh with Bob, he would go, "This marsh is just weird. It's just weird."
Oh man, this is only going to apply to those of us who go into marshes all the time. But like, I know exactly what you're talking about. Like, every marsh that I go into that doesn't look like the first marsh that I ever learned about marshes on, which was on Sapelo Island, I'm like, oh, this marsh is weird. Yeah, really weird. I'm glad that the legend Bob says that too.
Yes.
That's great. Okay, what is your favorite marsh plant?
I would actually have to say, Limonium.
Ooh.
Well, I would have to say it's a tie between Limonium and Salicornia, and only because it's so cool to watch those guys. Either when the Limonium is flowering, it just has the most beautiful little delicate blue flowers in this incredibly harsh environment, and then the Salicornia one, because you can eat it, and I don't agree that it tastes like a pickle because the vinegar's missing. But still, you can eat it if you need to. But then also the Salicornia is so cool to look at in the autumn when it senesces. It's like every single plant has a slightly different hue, ranging from like a dark ruby red to sort of a salmon to an orange, and it's so beautiful. So I love the Salicornia too.
I totally agree. Okay, what is your favorite marsh vertebrate?
Yeah, tough.
That's something that you never think about. Are those?
I know. I just like vertebrate, like, man. Probably the mamachogs. Yeah, probably the mamachogs. Yeah. So I mean, it's so cool whenever the tide comes up to see how many of them are out there. It's just insane. So, but I don't know. Also, the terrapins, they're super duper cute. So yeah.
Yeah, actually, I was just talking to somebody when where was I? Oh yeah, I was out on Nantucket this weekend, and I was talking to a herpetologist. And he was talking about the terrapins and stuff, because it's basically the terrapins have been like more or less, or at least functionally extinct from like southeastern marshes for as long as I've been down there. Of course, I mean maybe much longer. I think that they get caught up in fish traps or in I'm sorry, in shrimp nets a lot, right? But apparently the population's like pretty good up here in New England, especially like up in Maine and stuff, which I had no idea. I mean, even though I've been up here for for a few years now, I mean, I didn't know that. So yeah, apparently the terrapins are doing good. Do you see them at a relatively high frequency?
Oh yeah. Well,
really? So
the
cool.
main road going out to the marsh, we see them crossing it all the time. And they've built barricades to try and keep them out of traffic accidents. And they have like terrapin crossing signs all along the roads, like watch out for those guys. And you don't touch them. You're not supposed to touch them because I think they're all marked. They're all
really?
Yeah, they're all marked so they can watch their movement. So they're like, don't help them across the road. Just let them fend for themselves. But yeah, it's really cool because, I mean, we've even seen them burying their eggs before, like laying eggs. We saw a mother laying eggs one time. But
they do it in the marsh?
Well, there's like a sand right up next to where the road is, and she was laying her eggs in there. So those guys are very cool, and we see a lot of them actually out at the Tuckerton Marsh.
Wow. Yeah, that's really neat. I mean, in the whole 7 years that I worked in southeastern marshes, I found one.
Wow.
Yeah.
No, they're ridiculous at certain times of the year.
Wow. That's really, really cool. That's a great favorite marsh vertebrate. Really good one. Okay, this one will be really easy. Maybe this one will be harder. What's your favorite marsh invertebrate?
Oh, goodness.
Maybe that's harder, I guess.
Oh, that is hard. Can I have ties here?
Yeah. Well, okay. I think. Let me check. Yeah. We'll allow it.
Well, okay. So the hunting spider, Pardosa littoralis. I love those guys, and I love it especially. Like I think it's so cool. This generous predator that links not just the marine and terrestrial food webs, but also links the Spartina alterniflora and Peyton's food webs. Plus, wolf spiders are just so cool, and I don't know if folks have seen this, but I think people think of the spider world as a brutal one, but the mothers of these for the wolf spiders actually will carry their egg sacs around, and they seem to not feed a lot while they do that. They carry their egg sacs around, and then the babies all hatch out and climb onto mom's back and travel there for a while, which I think is super cool. So those guys are one of my favorites. And then we also have these really crazy Linnaeus spiders that have some of the males have the head modifications. And this is one of the ones that it actually looks like a tiny unicorn. And the eyes actually go around the little horn. Wow. And those guys, the Linnaeus, again, people think of, think of spiders and they don't think of all of the little loving things they do. Like some of these Linnaeus spiders actually give little nuptial gifts to the female using those modified head parts. And she actually lays more eggs when she gets that nuptial gift compared to when she doesn't.
Wow.
Yeah. So
what's the nuptial gift?
It's some sort of a secretion that apparently, I don't know what's in the secretion, but the secretion actually helps her lay more eggs. So
wow. Just when you thought that there was no love between spiders. I mean, I know we don't have a scientific definition of love, but if I've ever heard one, it's that it's giving a secretion to your mate. So that's love if I've ever heard of it.
I have to say too, the blue crabs are adorable, and the blue crabs and the fiddlers both. The fiddlers are hilarious, like the poor males that are just like, my claw, it's so hard to run around. But also I love it when the blue crabs come out because they just think they're so fierce and they cute.
Yeah, it's like as soon as, it doesn't matter how big they are, as soon as they're born, they're aggressive and they're like, hey, what's up? I'll get you. I can pinch you. I don't care. And, I mean, pretty much no matter what their size is, their pinch still does hurt. So
yeah. So, but not when it hits your rubber boot. So
exactly.
Like, oh, you're so cute. Look how cute you are.
That's exactly right. Cool. So what is your favorite? Well, we kind of maybe have gone over this already, but what is your favorite marsh life history story?
Oh, that's a hard one. Oh,
I know you have such a different suite of stories than most of the marsh ecologists I know, because you know so much about the arthropods in there. I mean, the spider nuptials is a pretty great life history.
Yeah. I'm trying to think if there's any cool parasitoids, parasitoid stories that I know with those guys out there, because those are always the most fun.
Yeah.
Parasites and parasitoids, and our parasitoids are, for the most part, a little boring. I mean, they just do their deal, but they don't have multiple different organisms involved in the life cycle. Yeah, I don't know. Probably the spiders and their mating habits.
Yeah.
No. Yeah.
Okay, let's see here. Who in the marsh do you think doesn't get their just desserts?
In terms of organisms?
Yeah. Yeah, I suppose you could pick a researcher, but no, organisms. I didn't say who. I should say what.
I would say, even though I think that I know David Johnson pays a lot of attention to them. I feel like the coffee bean snails need more attention. I think their role in decomposition is really important. And I think we need to know more about them. I think we need to know more about them in terms of where they belong in the food web, and who's eating them, and what sort of parasites do they undoubtedly have as a snail. I think that would be really interesting to get into in more depth. And I think we need to learn more about them, because they have a huge functional role on the marsh.
Yeah, no, I agree. I mean, and even they're like some of the more, they seem to be like active more year round than a lot of the other species. I mean, I was just in the marsh yesterday, and coffee bean snails are going crazy, decomposing still. You know, there's a lot of great stuff to decompose out there in the marsh right now. You know, I mean, I guess we're like late fall. I don't even know what this weather is. But yeah, but there. I mean, you know, I was just like,
up and down. Yeah.
I flipped if you know our listeners want to check out my Instagram at Hensel Pencil, they'll see that you know, I've got lots of pictures of just like, you know, like 50 coffee bean snails hanging out, you know, doing some decomposing in like a little area like this big. It's pretty.
Yeah, and I mean, this is their time, and a lot of those decomposers are out most of the winter, and I mean, our wolf spiders are too. As long as they get a sunny day, they'll be running around on the snow. So. Yeah, there is no, you know, rest for a predator.
Amen. Okay, last rapid fire question. What's the craziest thing you've ever seen in a marsh?
The craziest thing I've ever seen. Oh, good question. Performed by a researcher or by an organism?
I had thought organism, but researcher works as well.
Well, okay, I have to say, one of my favorite Bob Deno stories. So, and this, I actually wasn't present for this. I could just visualize it so well. So this is Shannon Murphy's story. So they were going out to Cape Hatteras. Shannon worked in Cape Hatteras for a number of years on the marsh out there. And they were going out with this, with a guy who, you know, basically was one of the parks directors around there. And he didn't seem to have as much of a sense of humor as Bob did. And he was like, be very careful of those branches, you know, they could fall on you and hurt you. And they were walking along, and all of a sudden Shannon was just like, where is Bob? Where did he go? And she went back to find him, and he had like dragged over one of the branches and was positioning it on himself, laying down like he had been struck by it. So the fun thing about Bob is he was always doing crazy stuff like that, you know, and especially out in the marsh and on the way up to the marsh and back from the marsh and anywhere else. And I just think that one of the reasons we just loved him so much is he's such a goof, you know. And he just, you know, he just made it, you know, the marsh, as you know, can be an unfun place to sample, you know, getting bit by greenheads, there's no shade, that sort of thing. But Bob always made it a fun trip.
Yeah. Yeah, I mean, in every poem or song that's ever been written about the marsh, you know, you have to rhyme it with harsh because the marsh is very, very harsh. And yeah, I mean, I've definitely found too that like working with great people sure does make the marsh a little bit less harsh.
Yes.
Well, that was the end of our rapid fire round, and that was the end of the questions that I have. It looks like it's been about an hour. Is there anything else you wanted to talk about, Gina, or that you wanted to touch on? I mean, we talked about a lot of your research that you have going on right now, which was kind of because it was going to be the other thing that I was going to ask about. But we've covered a lot.
Yeah. So I think the only other thing I was going to say is one of the things I really enjoy about being involved with salt marshes is everybody who works on salt marshes. It's not like the world's biggest group of folks. And we all seem to know each other, and everybody's really nice about, you can chat about, you know, different ideas with them. And it's not, you know, it's a friendly group of folks. And I think that's one of the other things that I just really enjoy about working in the salt marsh, is because you can start putting pieces together, and it is a complicated story because, yeah, it's only got 2 grasses and some forbs in there, but that marine terrestrial linkage is, you know, that brings a lot of diversity of interactions. And what's great to me is folks working in the different realms of the salt marsh talk to each other, exchange ideas. I really enjoy that aspect of it.
Yeah, no, you're totally right. I mean, the implications for being an intertidal system, but like having this crazy complex habitat is, really makes marshes so unique. Because I mean, you know, it's you know, very, very possible to spend a whole marsh career not even thinking about the things that you have, you know, you're making your whole marsh career about, or it's possible to think, you know, go a whole marsh career without doing the stuff that I do in a marsh. So it's like, and it's really cool that, like you said, you know, that we're able to start communicating with each other, so we can start trying to link those things. I mean, the research that you're doing, linking, you know, picking up the links and between those food webs is really, really cool, especially because, like, you know, like we've said a couple of times now so far, you know, it's like you look out and you think like, oh yeah, okay, you know, there's probably, you know, different things that live in different areas, but like, you know, they're also probably linked because, you know, it's just like one you know, marsh out there, and they must move back and forth. But like now we know, like, you know, stuff is so specific, and you know, so then even, you know, understanding the links between the terrestrial and the marine web is even more important when we now know that, like, there aren't that many of those links, right?
Yeah. So I think we assume a lot of things about salt marshes that tend to not be true. And it's really cool going through the history of where we came from and now what we think of it, because it was so dominated by the terrestrial part of it and the terrestrial ecologists, and not by the marine folks at first. And I'm glad that we're all getting on the same page.
Yep. I agree. Awesome. Yeah. Well, so I mean, if I could summarize up, you know, the big things that we've learned from Gina Wimp today. First off, are the importance of nutrients in these webs. You know, things really can be different. You know, the importance of of fragmentation, how separate these food webs can be. You know, the importance of spider nuptial gifts. I think, there's something that we've learned as well, and a great quote from Gina Wimp: "There's no rest for a predator." if it's warm, and it doesn't matter how much snow's on the marsh, the spiders have to go out and do their thing, huh?
That's right.
Awesome. All right, Gina. Well, thank you so much for
you're so welcome.
for joining. Really appreciate it. Cool. All right.